Good day, I'm Rogi van Staden. What causes tetanus? Spores of the tetanus bacteria, Clostridium tetani, are found in soil dust and animal feces. If these spores enter a deep flesh wound, the spores grow into a bacteria that can produce a powerful toxin, Tetanus spasmin, which impairs the nerves that control your muscles. Tetanus is a serious bacterial disease that affects the nervous system, leading to painful contractions and can interfere with an animal's ability to breathe, which can be life-threatening.
Hi, my name is Hector, and today I'll be talking about what are the characteristic features of tetanus. Now, the characteristic features are its gram-positive bacillus, it's rod-shaped, it's obligate anaerobes capable of producing endospores. The forming spores are terminally and subterminally. Most are motile with the peritoneous flagella, while others have a capsule like Clostridium perfringens. Moving on to the second slide. Given these features, can I explain the sudden onset of clinical signs? So the sudden onset of clinical signs would be jaw cramping, a sudden involuntary muscle tightening, which is often in the stomach, painful muscle stiffening all over the body, trouble swallowing, change in pressure and fast heart rate, and also cause seizures. Thank you.
Contraction and spread of tetanus. Tetanus is most often caused by Clostridium tetani, which is easily found in soil, droppings, or unhygienic places. Tetanus survives in the environment for a long time and is often passed on through wounds, particularly if the wound is unclean and open. Infection can also be acquired via the intestines after eating contaminated soil. In foals, infection can occur via the umbilicus. Tetanus is anaerobic and multiplies rapidly at the site of injury, producing a potent neurotoxin which can cause damage. In horses, the most common way for the tetanus bacteria to enter is through a wound, usually on the foot or lower leg, contaminated by the tetany spores in the soil. Tetanus can survive in the ground for a very long time. It will often survive with or without a host.
I'll be discussing the common portal entry of Clostridium tetanus and the samples to take to the laboratory. Clostridium tetanus is commonly present in the soil, dust, and the atmosphere. So it enters the body through the open wounds, bends, cuts, animal bites, and injuries caused by nails. Young animals can also contract this bacteria through surgical incisions when the instrument used to cut off the umbilical cord are contaminated. It does not enter the body through the mouth. The samples to take to the laboratory are tissue wounds, blood samples, and cerebrospinal fluid. The tissue samples are collected for histopathology testing. The blood samples can be grown in culture medium. McConkey or blood agar can be used, and then the cerebrospinal fluid is just analyzed in order to rule out other bacteria.
Hi, this is Bethany Moses. How to preserve and transport samples. One should transport the specimens in a transport medium. This needs to contain buffers and salts, as well as tissue culture, which can then add antibiotics, but it should not contain nutrients such as carbon or nitrogen. Another way to transport specimens is through anaerobic transport medium. This includes mineral salt-based, as well as semi-solid thioglycolate and cysteine, and this provides a reduced environment for the specimen.
My name is Debra Gordon, student number 152-65669. I did question number 10, which is to tabulate the differences and similarities between Clostridium tetani, other bacteria, viruses, and fungi in regards to classification, morphological characteristics, reproduction, genetic exchange, and laboratory identification techniques. Clostridium tetani are of the domain bacteria. They are prokaryotes and they are gram-positive pathogenic bacteria. Other bacteria are also prokaryotes but they can be gram-positive or gram-negative, whereas viruses are different because they are non-living cells, because they need a host cell to replicate in, and they are known as infectious obligate intracellular parasites. And fungi are eukaryotes and more complex, and they can be pathogenic. The morphological characteristics of Clostridium tetani are that they are rod-shaped and they resemble tennis rackets or drumsticks. Other bacteria have 5 different shapes in which they can be. Viruses contain a nucleic acid surrounded by a capsid, and they are smaller than bacteria and fungi. And fungi can be unicellular or multicellular, or they can be dimorphic, in which they have unicellular and multicellular stages in their life. The reproduction of bacteria, including Clostridium tetani, are that they reproduce asexually through binary fission. Viruses also reproduce asexually, but they do this through the host cell, and they insert their genetic material into the host cell. And fungi are very unique in that they can reproduce sexually and asexually, and they do this through a number of ways, but asexually through budding, binary fission, fragmentation, and sporulation, and sexually through sporulation. The genetic exchange of Clostridium tetani is lateral gene transfer, and it can be transduction, conjugation, and transformation. Bacteria also that is done through transduction, conjugation, and transformation. Viruses, they genetically exchange through transduction and fungi, they have horizontal gene transfer. Laboratory identification techniques of bacteria are usually gram staining to identify whether it's gram-positive or gram-negative, and then special stains to identify other structures or poor staining bacteria. Whereas Clostridium tetani, gram staining is unsuccessful, and so it's often done through toxigenicity testing. And viruses, they are identified through the polymerase chain reaction or immune electron microscopy. They are difficult to identify, so often clinical signs are used to identify. Fungi are stained using lactophenol cotton blue, methylene blue, or gram stain for yeasts. And that is a summary of the similarities and differences between Clostridium tetani, bacteria in general, viruses, and fungi.
Hi, my name is Leoni Botha, and my student number is 161-46532. The tetanus vaccine is also known as a tetanus toxoid. It is an inactive vaccine, which means that the disease-causing microbes in these vaccines have been killed prior to making the vaccine. Once killed, the microbes cannot revert back to their disease-causing state. The dead or weakened version of the disease enters the body, causing an immune response, which leads to the production of antibodies. A unique feature of this vaccine is that antigens are released for several months because of its ability to form a long-term depot. Through routine vaccination, the disease can be effectively prevented and is usually administered yearly or as indicated.